Dr. Marjan Assefi, CEO of the Marie Curie Science Research Center, said, "Why does cancer, despite all the advancements in medical science, still remain such a formidable adversary?" Indeed, in the quest for understanding and conquering this complex disease, scientists have made extraordinary strides. Yet, cancer continues to be a leading cause of death worldwide. But what if we could rewrite the genetic code of cancer cells, turning off their destructive potential? Enter the world of genetic editing techniques, a realm where science fiction meets reality. In the late 20th century, the idea of manipulating genetic material was a mere dream. But then, in the early 90s, came the first wave of genetic editing techniques, spearheaded by the discovery of zinc finger nucleases, or ZFNs. These are proteins that can be engineered to bind to specific DNA sequences. Once bound, they can introduce breaks in the DNA strands, allowing scientists to remove, add, or replace genetic material. Then, in the first decade of the 21st century, a new technique emerged known as transcription activator-like effector nucleases, or TALENs. Like ZFNs, TALENs can be customized to bind to specific DNA sequences. However, they are generally considered more efficient and versatile than their predecessors. Moving forward, in the second decade of the 21st century, a revolutionary technique known as CRISPR-Cas9 took center stage. This tool, borrowed from bacteria, allows for precise and efficient editing of genomes on a scale never before possible. It's a cut-and-paste mechanism that can remove unwanted genes and insert desired ones with unprecedented accuracy. Dr. Assefi added, "These genetic editing techniques, each more powerful than the last, have opened up a new frontier in cancer treatment. By enabling precise manipulation of genetic material, they provide an avenue for targeting the very roots of cancer, the genetic mutations that give rise to the disease. ZFNs, with their ability to introduce breaks in DNA strands, have the potential to disable cancer-causing genes. TALENs, more efficient and versatile, could not only neutralize harmful genes but also introduce beneficial ones. And then there's CRISPR-Cas9, the most precise and efficient of them all. With this tool, scientists could theoretically reprogram cancer cells, turning off their ability to grow and divide uncontrollably." Dr. Assefi added, "Yet, as promising as these techniques are, they also raise ethical and safety concerns. The power to edit our genetic material is a double-edged sword. On one hand, it could lead to cures for previously untreatable diseases. On the other hand, it could potentially be misused, leading to unforeseen consequences. In the end, while the journey to conquer cancer remains a challenging one, the development of genetic editing techniques has provided new hope. These tools, in the hands of skilled and ethical scientists, have the potential to revolutionize cancer treatment, offering a future where a cancer diagnosis is no longer a death sentence, but a problem that can be solved at the genetic level. The quest continues, and every step brings us closer to that elusive victory.